So, question 3. A student made a squash preparation of a root tip to observe the stages of mitosis. The student used the following statements to identify cells in metaphase. Pairs of homologous chromosomes lined up along the equator of each cell, crossing over taking place, chromatids visible. The number of correct statements about metaphase and root tip cells is, well, let's see. Metaphase in mitosis would look like this, with the chromosomes lined up along the equator and spindle fibers getting ready to attach. Well, let's see. First statement, pairs of homologous chromosomes are lined up along the equator of each cell. Well, here we do have chromosomes being lined up along the equator. However, they're saying pairs of homologous chromosomes. So that would look like this. This is only seen in meiosis, where you have pairs of homologous chromosomes. In metaphase, you do not have pairs. You only have the chromosomes. And let's see, so that is incorrect. Crossing over taking place. Again, crossing over only takes place in meiosis because they only occur with pairs of homologous chromosomes. Because what happens is you've got two chromosomes and they cross over at a point. So if we go for this point, this is the end result. As you can see, they've switched over. And that's just increased genetic variation. This is not going to happen here, so that's crossed out. Chromatids visible. Technically, yes, that is right. Because even though people generally say chromosomes and chromatids interchangeably, they're not. Technically, this is a chromosome and this is a chromatid. So technically what we have here is chromatids, and one is called a chromosome. So here, the number of correct statements is only 1, and so the answer is B. The diagram shows the method that the students used. A root tip of length 2 centimeters was cut from the plant. The root tip was placed in an appropriate stain and dilute acid and left at room temperature for 10 minutes. The root tip was put onto a microscope slide and a cover slip placed on top. The cover slip was gently pressed down. The preparation was observed using a light microscope. The student was disappointed with the slide that had been prepared because the nuclei were poorly stained and no stages of mitosis could be seen. Explain why the student had to make sure that an appropriate stain was used. Well, this experiment would have been done as part of the lab work that that the school would need to submit to Edexcel to show that you've passed. So, starting, the question says, explain why the student had to make sure that an appropriate stain was used. Well, why do we stain? Well, we stain so we can see the chromosomes. Because normally, the chromosomes are not visible. Well, yes, not visible. And so then they say, why must an appropriate stain be used? Well, not any stain can be used. Most likely an acetic also stain would be used. And why? Because that would attach to the chromosome. And that's the bit that we want to see. If you use a stain that didn't attach to the chromosome, you would still not see the chromosome. And it's also acceptable to say it attaches to the DNA, because the DNA is in the chromosomes, and so the chromosome will be visible. It's also appropriate to say attached to the histone, because histones are proteins that are the lengths of the DNA are coiled around. And so if you stain the histones, you would see the chromosomes. Now, Describe the changes that need to be made to this method to allow stages of mitosis to be seen. Well, firstly, they used a length of 2 centimeters. If you'd done this practical, it would have said used 5 millimeters to 10 millimeters, or you can put a shorter length. Why? Because even though in this question you don't have to give an explanation, because all they want is a description, because the question says describe, I'm going to explain why you would do these changes. So if you use a shorter piece, you're cutting the root tip that's younger and that has newly emerged. And so that will produce the best results. Now, another thing you could have done is they stained and diluted the acid at the same time. What you could do is you can put an acid first. Why? Because you put in acid so that the cells break open. And so after, when you put the stain will find it easier to enter and attach the chromosomes, because the cell has broken up. Another thing is that you can heat the root tip in the acid. And that, again, is to help it break the cells open more. Both these steps can be done, again, with the stain instead. And that would just be to intensify the stain. You can, before adding the stain, you can tease the piece that you cut apart. And you can use that with a needle. And that's to help break open the cell. Again, making it easier for the dye to enter. And lastly, you can mention to warm the stain, so you can intensify the stain. And this is done after you've gotten a cover slip and you've put in a sample, you've got on a slide and you've put in a sample and you put the cover slip on. And what you can do is you can wrap it in a piece of tissue and you can gently press that down. And so that's squashing it. After you've squashed it, and that's just to help to spread the cells a bit more. Same thing with teasing it, and helps it spread a bit more and helps just to make things a bit better. And that would be it. This is on the Edexcel website, the whole practical and the reasons why they've also done it. So that includes, seeing things like maceration, which is just to break up, and has the whole practical. So I will put a link up in the description if you want to read the whole practical and what they used for it.